Good morning, everyone, and a warm welcome to this Baltic Breakfast on this lovely December morning. And welcome to you who are following us online. Today we are gearing up for the COP15, which is really exciting, only a couple of weeks ahead. With me today I have really some excellent researchers, Laikre and Susa Niiranen, who are from Stockholm University, who will help us to give some of the science behind. My name is Ellen Bruno. I work at the Baltic Sea Centre at Stockholm University. And at the center we do research, but we also communicate and we try to follow the political processes going on and to make sure that the decisions that are taken are based on scientific knowledge. And one of our tools are these Baltic Breakfasts. So we will have two 50-minute lectures, and then we will have questions for 50 minutes. So save your questions for the last 50 minutes. And for those of you who follow online, you can write your questions, and please follow the instructions on the screen, because I think there are some specific ways of doing that. And I think with that, we'll just start to welcome Susa first. And Susa, you are here today instead of Robert, because Robert Blasiak, he had an accident actually. He fell yesterday. He's okay, it was his leg. But so kindly you're jumping in and you're taking his slides. So we know that this is not your expertise. You are actually expert in how different factors affect ecosystems, right? So anthropogenic pressure and also natural pressure, of course. Yeah. But yeah, I think you can manage this. I think you worked quite a lot with Robert. Good. So a warm welcome to you.
Thank you.
Charles. I think, so you can stand here where I stood.
I was told to stand here.
Okay.
No? Yeah?
I'll move.
Okay. Is det okej? Ja. Det är det. Ja, perfekt. Så vi bara har en presentation mode så då funkar det bra. Bra. Ja. So thank you very much, Ellen. Good morning to everybody. As Ellen said, I'm a stand-in for Robert Blasiak, my colleague. And with Robert together, we lead the Human Oceans research team at the Stockholm Resilience Centre. What you're going to see today, it's really Robert's content, Robert's thoughts, but I give the voice to them today. And the talk is going to be about the marine protected areas and their contribution to achieving biodiversity and climate targets. So, of course, Ellen talked about the COP15 that is starting in a couple of days, and the Convention on Biological Diversity is really at the center of this. And this was established approximately 30 years ago, 92, or maybe quite exactly 30 years ago, actually. And it has currently approximately 200 state parties. And it has been ratified by all UN member states, with the exception of USA. There's three main goals. The first one is the conservation of the biological diversity, so the very core of it. The second one is the sustainable use of this biological diversity, so the benefits it gives to people. And the third one is the fair and equitable sharing of benefits arising from genetic resources. So if we then look a bit about the past, present and future of the CBD, we go for the past, and we had this strategic plan for biodiversity for 2011 to 2020. And this was a global 10-year framework for action by all countries and stakeholders. And the aim here was really to save biodiversity and enhance its benefits for people. And it had 5 broad strategic goals, and then 20 a bit more specific so-called IKE targets. So present, we know this was the past decade we were working on this. So what happened then? And now we have the kind of facet in our hands. And this is the paper, it's a news piece from Guardian 2 years ago. And there were titles like this: World fails to meet a single target to stop destruction of nature, according to UN report. And it seems quite dark, but there are good things also. So yes, we didn't meet the targets exactly, but there was good progress going on, and these things were really lifted up. So if we go to the future then, and more specific to the COP15 that's coming up, and this has actually been delayed 4 times due to COVID. And it was supposed to take place 2020 originally in Kunming, China. And now it will be held in Montreal, and it will start in 8 days, if Robert had the count right. I think he did. And this should kind of result in this kind of renewed, revamped, post-2020 biodiversity framework. So you may ask then, where is ocean conservation in this CBD? And it really is there. It's the IKE target 11. It's lumped together with the terrestrial and inland waters, but it does clearly say that by 2020, 2 years ago, 10% of coastal and marine areas, especially areas of particular importance for biodiversity and ecosystem services, so we are talking about the benefits the ocean gives to people, are conserved through effectively and equitably managed, ecologically representative and well-connected systems of protected areas, and other effective area-based conservation measures, and integrated into wider landscapes, and particularly for our interest, the seascapes. So if we take a look at what the situation is today, so these statistics, it's actually from this month, November, it's still the last day, 2022, and we see that 8.5% of the ocean is protected. And why we say protected is that there are different levels of protection when we talk about MPAs. And if we take a look at this map, and we see the dark blue regions here, it's only 2.1% that are fully or highly protected of these MPAs. And many of these areas actually that are highly protected are in very remote regions where we would not really have this human influence on them in any case. And I work with people who work with the Baltic Sea. It's a Baltic Sea breakfast with the Baltic Health Index. And I asked, what is the number for the Baltic Sea? And that number was between 13 to 14% now. And there is the same stories there going on, that you have different levels of protection of these MPAs. So where to go next? There's an advocacy going on. And it's 30 by 30, a very catchy slogan, I would say. And this means that 30% of world's oceans should be conserved by 2030. So we have approximately 7-8 years for that. And this campaign was launched 2018. And if you think that approximately 200 countries signed the CBD, now 128 countries have already committed to this 30 by 30. And you see a map here. And if you zoom on the Baltic Sea region, for example, which I think is interesting to you guys, we see that Finland, Sweden, Denmark, and Estonia have committed to this so far. But is there science behind this? Is it just a catchy slogan, 30 by 30? There is. So in 2009, there was a meta-analysis published of 33 studies focusing on fisheries. And they concluded that 30% is actually the optimal area to protect in the oceans in order to gain benefits for fisheries. 2016, there was a meta-analysis now with 144 studies and had a bit broader scope. So it was not only fisheries that was looked into. And their number here is 37%. So the science says approximately 30 to 40% should be protected. So then, if we talk about the meeting coming up, there's a first draft of this kind of what I say revamped post-2020 global biodiversity framework. And there's 21 draft targets now. And once again, there is one that includes the ocean. And it does really say that at least 30% of now land and sea areas globally should be conserved through, once again, effective, equitably managed, ecological representative, and well-connected systems of protected areas, so MPAs. So then Robert asks, you may ask, why do we push so hard for the 30% if the 10% target was never met? So we know now we were at 8.1%. There really is urgency of action. So we know that the stressors on our global oceans, they are not decreasing, it's the opposite. And also the science says that MPAs provide conservation benefits. That's clear now. Our experience says that. But there is another side of the coin. So now it's increasingly promoted that the MPAs really can be an ocean-based solution for climate change mitigation as well. And that's not the only thing the MPAs can provide. So we, of course, have the biodiversity conservation, fisheries benefits, sustainable blue economy on several sectors, decreased risk of extreme events, increased resilience and CO2 sequestration. So there's the carbon harvesting that mitigates climate change, preservation of cultural values, for example, small-scale fisheries, better ocean knowledge, and even better ocean governance. So then again, Robert is asking, or they were a year ago, where is the evidence in all this? And they did now do a systematic literature review of not 200, but 22,403 publications. And these covered 241 different MPAs globally. And here they really focused also on question of the climate change mitigation and adaptation, among other things. And what they did find, it was very clear. There were clear climate benefits of the MPAs. There were also other social ecological benefits, like coastal protection, benefits for fisheries. But the outcomes, really, and the benefits, they depended on the level of the protection. So this also touches a bit what I showed you on the map earlier. You have MPAs, but they can be minimally protected, or they can be fully protected, and everything in between. And they're still called and classified as MPAs. And what they found out in this paper, Robert and colleagues, was that if these MPAs are fully protected or highly protected, you really get these broad range of benefits. But if they are not, the benefits really are not there. So these are the three key findings and take-home messages that Robert and myself would like you to take home from this. The first one is that there is a clear signal that the protection level should be prioritized in MPA design. There is really a risk that this 30 by 30 target leads to loads of weakly implemented MPAs with limited benefits. So it could look good on paper, but the effects are not really there. The second thing is that the governance, it's a fundamental factor in determining whether MPAs will succeed. And this means that you should have diverse stakeholder groups that would be consulted and included throughout the planning and implementation process. So this is the only way you get the legitimacy for this. And there's also a considerable scope to expand the consideration and recognition of MPAs in national climate strategies. And this should be looked further into. So that's all I had. Robert sends his wishes. He was really regretting he could not be here. But he also said that if you have any specific questions, you want to have dialogue with him, please contact him. I think the information is on the website of the event. Thank you.
And thank you, Susa.
Thanks.
So I have one question for you, because I know you're also an expert on it. So you say that more protected areas are more efficient also for storing carbon dioxide and climate gases. Do you know, is there like specific mechanism that needs to be withdrawn from marine protected areas for that to happen? Like, is it the fishing or is it the exploitation? Or what do you know what?
So basically, if you store carbon in these systems, we count, and I think Robert and the colleagues counted also, that if you store it for 100 years, the carbon in the system, then you basically, it's out of the climate cycle. Okay. So then we are talking about things like kelp forests, seaweed beds, macroalgae, but we're also talking about fish. That so fish, when it dies, it sinks down, and if you have the deep ocean, the carbon stays there. But it's a bit debated how large proportion of that is actually, like how relevant that is for the climate change mitigation.
Okay, thank you.
Yeah.
And Linda, your turn. And now we're gonna move to a little bit of a different area and a different target, actually. And Linda, you're a professor at population genetics. Population Department of Zoology. And yeah, you're going to take us through some genetics, which is also linked to the ocean, of course. So take it away.
Okay, can you hear me?
Not really.
Thank you. I think it's better now.
Yeah.
Okay, thank you very much for this opportunity. Very happy to be here. Most people, when they think about biological diversity, they think about species diversity and about ecosystems, knowing that we have variation and in different species and how they interact, and this is really important. But in addition to this level, and we also know that when specific species goes extinct, this can have effects on entire ecosystems. But in addition to these levels, we also have variation within species, genetic diversity. And this diversity originates from the DNA level, where a specific gene can occur in different variants. We call them alleles, and I symbolize them with colors here. So individuals can be genetically different, and that builds up into genetic variation within populations and between populations. And between population genetic diversity often reflects adaptations to local environmental conditions. And this variation is actually the key for evolution. Here lies the resources that can be used for adaptation to rapidly changing environments. And when a specific genetically distinct population goes extinct, this can actually have similar effects as when we lose entire species. It can affect other species and ecosystems. So loss of genetic diversity is a large problem and a level of diversity that's really, really important. And we know from decades of conservation genetic research that when we have lots of genetic diversity, to the left there, and I symbolize again genetic variation with colors, this is often associated with high adaptive capacity, good potential for long-term survival, and high resilience. Whereas the opposite goes for populations with low genetic diversity. They often have low adaptive capacity and is weaker potential for long-term survival. And there are many examples of these things, and one recent scientific example comes from corals, from these workers in 2019, where they have seen that corals were able to adapt to warmer ocean temperatures depending on particular genotypic compositions. So genetic variation within species of corals affected their ability to adapt and survive. And a recent example for depleted population and the negative effects is wolves on Isle Royale in Lake Michigan in North America. So this population has been isolated on this island for 6 decades, and were long regarded as an example that genetics was not so important. But then what happened was just what conservation genetics warn about, that as genetic diversity is lost over generations, as inbreeding accumulates, this has negative effects. And inbreeding depression, spinal disorder resulted in a collapse of this entire population. And there are other examples. Here's another recent examples from another habitat-forming species, kelp forests, in the oceans outside of Australia, where low genetic diversity areas, again, were more susceptible to a marine heat wave, whereas high genetically diverse kelp forests were much better at surviving the heat wave. From our own areas, eelgrass, similar kind of connections have been demonstrated there. As we increase genetic diversity on the x-axis of the eelgrass, you see the graphs are going up. Other species increase in their occurrence, biomass and so forth increase. And genetic diversity can help individuals, species, populations to adapt. And the recent examples, again from a marine environment, killifish outside of North America, some highly polluted areas, and it turned out that standing genetic variation, existing variation within populations, allow them to adapt to really polluted areas that were toxic to the original population. And we have many examples from the Baltic Sea. Many marine species have adapted to the particular environment of the Baltic Sea, and herring is one where Leif Andersson's group in Uppsala have demonstrated beautifully how specific genes leads to adaptations. For instance, rhodopsin that makes the herring possible to have better vision within the Baltic Sea environment. And more examples exist on this. And the conventional biological diversity, already from the start, it recognized these three levels of biodiversity: ecosystem, species, and genetic diversity. So that's in the convention text. But when it comes to implementation, genetic diversity has long lagged behind. Not much has been done. The text is there, but nothing is really done. We have not really had any targets or ways to monitor this level of diversity. And this has been pointed out repeatedly by scientists, warning about the problems of neglecting genetic diversity. And so we feel now, conservation genetics researchers, that improvement are urgently needed, and we hope there is a chance for improvements at COP15. So researchers in conservation genetics from all around the world have gathered together, formed the Coalition for Conservation Genetics. So these are people from the IUCN, experts in genetic diversity, Society for Conservation Biology Working Group, GeoBon, and the EU-funded network GBIKE on genomics and the importance of genomics for ecosystem functions. All of these groups have joined forces to try to provide advice, provide information to the policymakers for COP, and looking forward to a new framework where genetic diversity is included. So we really need a strong goal and target that includes all species, including wild species. But we also need means to measure change. So we need indicators. And factors that can result in loss of genetic diversity is if we lose entire populations that are genetically distinct, or if populations become too small, as we have already mentioned. These are the two main processes that result in loss of genetic diversity. But we also can have human-induced harvest, when we harvest natural populations through fisheries, for instance, and if the individuals that we removed, if they have specific genetic compositions, these kind of processes can result in loss of genetic diversity. And finally, if we have a natural population and then we start to release something that has a different genetic composition into the nature, that's done very much in fisheries, for instance, this can also result in loss of genetic diversity. So these are things we want to keep track of. And this network, Coalition for Conservation Genetics, propose indicators for these different levels, particularly indicator 1 focusing on within species diversity. So these 3 indicators have now been the first indicator that focuses on within population genetic diversity. This is now included and proposed as a headline indicator in the monitoring framework, and we are really happy about this and really hope that it will remain there. And this indicator relates to a scientific, well-established rule of thumb saying that when we have a genetically effective size, and that's a traditional population genetics parameter that measure the rate of genetic loss, when we reach NE 500 and above, then we have a potential for long-term survival. We have a good adaptive potential in such populations. So that's why we focus this indicator on that target. We also think we need to focus on maintaining the proportion of populations maintained, so that looking at between population diversity. And the third indicator is use of DNA-based methods. Many nations cannot do that, so the first 2 can be used by proxies. And Sweden is already implementing some of these work. So the Swedish Agency for Marine and Water Management and the Swedish Environmental Protection Agency, together with researchers at several universities, have already initiated monitoring programs in Sweden. And one of the species that is at focus is the herring of the Baltic Sea, in particular because here managers want to find out how many genetically distinct spawning populations do we have along the coast. So we have, over the past 2 years, carried out this huge sampling effort, thanks to coastal fishermen who have provided the samples. And now they are off to be genotyped at a laboratory to try to answer these questions. Also, to what extent separate populations are fished, if there are genetic effects, and if there is temporal stability. So in some instances here, the arrows here shows where we have samples from the past, so we can see if we have temporal trends over time. And similar work is now also ongoing for cod, eelgrass, bladderwrack, brown trout, and the salmon. And more information, if you want to know more about these efforts, you can find a lot on the net. The Coalition for Conservation Genetics has a website, and we will also be at COP15, have a side event there. And G-Bike will also be at COP15 on the EU network. And in Sweden, we have a site called BaltGene, where you can find more about genetic diversity of Baltic Sea species, Swedish Environmental Protection Agency, and some other resources. Thank you very much for your attention.
Wow, thank you. I think we can close this. And if you can both join me at, I don't know where it is. Voila. So we have some time for questions. Come. Thank you, Linda. I'll start with you. And I have a question, of course. So you're saying that Sweden is already trying to implement this monitoring system, or are doing that, and that they have a couple of species that are selected. How were they selected? Like, why did you choose those? Or why did they choose them?
Yeah, it was a collaboration between managers and scientists. So these are species that the Swedish Agency for Marine and Water Management, Havs- och vattenmyndigheten, they regard them as specifically important. They had management questions relating to these species. For instance, the herring situation. So that's how they were selected. And the idea is to have, we have actually 12 species that they wanted to focus on initially. And so some of them are started. We are starting in small steps, and will continuously improve this effort.
Okay, so there will be more species eventually.
Yeah, and there are also some terrestrial species that the Environmental Protection Agency looks at. For instance, pollinating species.
Oh, cool. Just going to say, if you have questions, Jenny will run around with a mic. So just raise your hand. So you're saying that there has been a target on genetics before, but it was only on domesticated species, right?
Yeah, it was target 13, Aichi target 13, that focuses specifically on domesticated species, socially, economically valuable species, also their wild relatives, but very little on wild species broadly. And the main indicator used was the number of domestic breeds that were maintained. And of course, domestic breeds, that's also very important, but misses out completely on what's happening to the wild species. So that's why what we are highlighting now, that it's extremely important to also include wild species in the upcoming framework. Mm.
And do you know why wild species weren't included? Were they not important enough, or did we don't have the technique, or?
I don't really know. I mean, genetic diversity has long lagged behind, and that's been highlighted, like I said, by many. And I think there are many complex answers to the question. People don't really see genetic diversity. It's not so obvious that it exists. It takes time before the effects of genetic diversity is shown, and you have the image that you definitely need DNA techniques to do anything, to understand anything about it. And those techniques have been expensive, but they are becoming much more cheap now. And it also becomes, people are more knowledgeable about it, I think. So that I hope it's going to change at this next COP.
Cool. Yeah, Gun? Oh, sorry. I have one there first.
Thank you, Lina.
Technique.
Hello. Thank you, Linda, for a very good presentation. Also, Susan. I have a question to Linda, and that is, we have these all local adaptations of fishes, for example, the herring in the Baltic Sea. But what if some of these populations are lost? I mean, the local, the whole subpopulations, for example, can they be can they be reinvented by mother population again? Or, I mean, if local population are lost, can they be re-established from a mother population?
We don't really know that, but it's definitely going to take a very long time. So that's why it's so important to maintain populations as much as possible. It will take evolution a really long time to create them again, and perhaps it won't even be possible. It depends on what kind of genes or what variants are available in remaining populations, and if they represent genes that can adapt to a particular environment. So that's why why we think it's important to focus on maintaining populations. And that's often now disregarded in, for instance, red list processes. But we are working and see how we can use red list data and improve the process for red listing. That's another thing that's ongoing now with Artdatabanken here in Sweden and many other places around the world. So it's really important to maintain populations because we don't know if we can recreate them.
Thank you.
Hi, thanks a lot, both of you, for very nice presentations. And I have a question for actually both of you, MPAs and genetic diversity. Are you sort of in a positive mood for the upcoming CBD? Will we see change?
Do you want to start?
I can start, but I'm Finnish. I'm hardly ever in positive mood. But but to be honest, I don't know enough. So my gut feeling is that not super optimistic, and I think it's mainly this thing that it's the different levels of protection that we are still missing. So it looks good on paper, this 30 by 30 and all this, but we see it from the Baltic Sea, for example, as I said earlier, that it really makes a difference what level of the protection the area has, and it's not really, highly protected and well managed areas. It's not all of them, nearly. So.
Yeah, I feel both ways. I mean, with respect to genetic diversity, I see that finally we have some wording on the table that are an immense improvement from before. That's largely, I would say, because of the work that we have been doing over the past 2 years. We formed this coalition, and then we have repeatedly had webinars, provided advice, making statements. And along the way, the text has improved for the goal and the target that relates to genetic diversity. But we haven't seen the final outcome yet. So you never know until you are there what's going to happen. But right now, it looks relatively good. But improvements can be made, and we provide those advice constantly to the national focal points. So I'm hoping for genetic diversity that we will have something that's better than before, at least.
We have a question from our digital audience. How do you find OECMs in the protected area targets, threats or possibility? And as I Googled this OECMs, it means other effective area-based conservation measures. But perhaps this could be
I can't answer that. I'm super sorry. But please do contact Robert. He's very knowledgeable in this. And he's very happy to answer your questions. So send him an email, call him. I'm sure you will get the answer. And thanks for the question.
Hey. First, thank you very much for 2 very interesting talks. And I have a question for Linda. Some species were mentioned for monitoring of the genetic diversity, but I heard nothing about microorganisms. Is genetic diversity within microorganisms something that has been looked at in the past, or something that we are interested in looking at in the future?
Yeah, it's definitely interesting. Not so much has been done, but there is an interest there, and there are people working on it. So definitely that's an area of interest, and that's an area that's becoming more and more recognized.
Hello, Thomas Vikander from the Council of Baltic Sea States. I have a question for you. about the marine protected areas, and how much do these areas protect the fish stocks if we stay in the Baltic Sea? How much, do they protect the fish in the water?
It really depends on the MPA and how it's managed. And I got the numbers yesterday from this Baltic Health Index team, but I can't remember the percentage of these. I know there is, from the Baltic Sea Centre, you have an expert here, don't you? Maybe, so maybe you guys can have a discussion afterwards. That'd be great, yeah.
Thank you.
Sophia, yeah.
One thing when it comes to genetic diversity and MPAs is that we did a study a few years ago looking at is genetic diversity included in the MPA management plans, and there were nothing there. So we have a problem also with that we have national guidelines for genetic diversity, but when it comes down to MPAs, for instance, there is no text or how genetics can be is considered. So just wanted to add that to your question. Thanks. Sorry.
Thank you very much. My name is Henrik Svedeng, coming from Baltic Sea Centre. Actually, I have two questions, but I start with protected areas. Nearly half of the surface of Earth belongs to the high seas. That is, nobody owns it. And does this 30 by 30 include the high seas, or are they left out?
This is what I understood, but maybe somebody who's more knowledgeable about this can correct me. Sophia, do you know?
I think they are included.
Yeah.
I think they are included, yes.
Yeah. Thank you.
I think most of them should be.
Yeah.
Actually.
But it's tricky, because that is the tricky part. And also when it comes to all, we have talked about the pelagial a lot today, but when it comes to, for example, the resources that are in the seabed, I think in the future that's going to be one of the key questions also.
Right. And I have a question to Linda as well. A recent paper on genetics in herring, on historical development of, or the erosion of genetic diversity in Baltic herring has been going on for a very long time. Is the target for genetic diversity conservative enough?
We hope so. We hope so. But there is definitely room for. I mean, we have this threshold value, effective size 500. Don't go below it. But that doesn't mean we should reduce populations. We need really large populations. We know that when really large populations are declining, we lose allele variants rapidly. So that's really a minimum threshold and that we should not go below. So it's extremely important to maintain large populations.
Okay, I'll just grab a question for you, Linda. Okay. So you said that the indicator 1 will probably be included, and the other 2 will be more or less voluntarily, if member states want to use them. Do you think, why is that? Is it because we don't have enough knowledge, or what's the problem? Why don't we include all 3 indicators if they're needed?
It's a policy process within the CBD. So they have for the new framework they have organized them into headline indicators. Those indicate they were not there before. So this is a new thing for the CBD, and they are supposed to be. All nations need to report back on the headline indicators. So there is a pressure for making them few. That nations don't want. Some nations don't want so many. And there are 5 goal A that deals with biological diversity convention, ecosystem, species, genetic diversity. There are all 5 headline indicators there. So there's argumentation for reducing the number. So it's not certain that this indicator remains. So that's still a struggle to get it in there. And then the component and complementary indicators are lower levels that nations can use if they can and want to. So that's obviously for some nations these will be very helpful. Hopefully I mean, I look forward to, if we have at least one indicator, if we are starting monitoring genetic diversity, this will result in results coming forward, that we finally get some more data. We have a relatively little monitoring data yet. So I'm hoping that it will at least start and then go forward from there. So that's my hope.
So we get the monitoring in place.
Yeah.
Yes.
Hi, my name is Per Larsson, and I got a question concerning the Swedish position at COP15. Do you know in what extent the Swedish government has consulted or involved academia and researchers in forming the Swedish position?
I'm not fully certain. I mean, we have a new government now. I'm not exactly sure what has happened over this short period of time. Right now, Sweden is not speaking on its own. It's the EU. It's EU coordination at this level. So at meetings below, substa meetings, for instance, then nations speak on their own. But here, it's EU coordination. And the EU delegation for Goal A, which I am focused on because it's there, genetic diversity that's led by Torbjörn Ebenhardt from Uppsala, so he's going to lead the EU delegation. And I know he has a very strong opinion on making a strong goal. He definitely supports the genetic diversity inclusion. We have a video of him. If you go to the coalition webpage, you can see several national focal points supporting our suggestion. And Torbjörn Ebenhardt leading the EU delegation is one of them. So that's a good thing.
Yeah, I can just add that as a university, we've been involved as well in these interest groups that they have. And one of the things that they keep stressing is that Sweden are really lifting that they need to be science-based, the targets. And for example, there was some idea about the targets for 2030 being lower than the target for 2050, like you would do for marine protected areas, that you would have one target and then another more ambitious target, and that would be the same for genetic diversity, which would of course not be very smart. So I know that they are working really heavily on the genetic diversity part, at least.
Yeah, and always along the road of CBD, there's been a strong influence by scientists in the process. So we have had the Swedish Council for Biological, the Swedish Scientific Council for Biological Diversity, based at Naturvårdsverket right now, gathering lots of scientists in Sweden on biodiversity knowledge, making recommendation into the process. So I think it has been over the years good here in Sweden with that.
Hello, my name is Roland Fredriksson. I was from the map. I think it was you. It was Sweden, Finland, and Estonia in this.
And Denmark.
Yeah, and Denmark. Yeah.
Yeah.
Why not the other country? Why do you know? Why?
I would assume it's a political thing. Like I think these things are not so easy. For example, in countries like Germany, I think we've seen it in the Baltic Sea region also. Otherwise, that it takes quite a lot of time, and there's different interests. But maybe there's probably more people like Bo Gustafsson who can speak about this within the HELCOM, not MPAs, but within the HELCOM context. But that's my understanding, at least. For this specific process, I'm not quite sure.
It should be in their interest, I think.
It should be, but they are big and they are quite complex with the management.
So thank you, everyone. We need to wrap up. I'm going to have one last question. So what would make you very, very happy if it came out from the COP15 in a month?
I sound like a broken record, and it's part of Robert's broken record also, but it's that it's not only that we have the 30 by 30 or 30 by 50 if it's going to be that, but really it matters what type of protection, and it has to be very carefully planned. So maybe in some cases it's better that you do less, but you do it in a meaningful way, so you just don't get the stamp.
All right. No paper parks.
No.
All right.
And I would be very happy with a goal that recognizes genetic diversity of both domestic, much we need to maintain over time, and the target that also stipulates that we need population diversity with a quantitative target, saying how soon, up to 2030, we need to have strategies monitoring program in place, to have strategies into place, that we need to start to do some. Indicator remains, and that the other indicators are in there as well. That's my hope. Then I will be really, really happy.
Cool. Thank you. And I want to take the opportunity to say that the next Baltic Breakfast will be on the 15th of December, and it's about river-based pollutants and how the Water Framework Directive is actually protecting our rivers from pollutants. And if you have more questions, and if we didn't have the time to answer them, please send them in, and we will definitely try to answer them on the webpage afterwards. And thank you all for coming. And the researcher will stick around if you have more questions. And now all of you go out and save the world, because I know you're all heroes. And thank you for today.
Thank you.
Thank you.